Hello, my gentle and of course very modern apes. If I sound a little sick, it's because I am. I have had this cold that I cannot kick for like a week now, but that is not the reason why you've seen sparse content from me lately. I'm working on a very big project for the channel. It's a very big video. It's a very long video, and that's coming from me. So you know it's actually very long indeed. I hope you'll like it when it comes out. It's going to be within the next week or so, look forward to that. But in the meantime, like a lot of cool stuff has happened within biological anthropology, and I figured that I would just hop on real quick and kind of kill two birds with one stone here. I'm going to be appeasing the algorithm gods so they don't think that I've vanished off the face of the earth and, shadow ban my channel or whatever. And I'm also going to get to give you my opinion on some cool recent discoveries within biological anthropology. For those of you who are biological anthropologists and you've discovered really cool stuff recently that I'm not covering right now, please keep in mind that I am extremely biased in the stuff that I find exceptionally interesting. This is a subjective list, and really it's only in list format because YouTube loves lists. What this actually is just a series of cool studies that came out recently, and I think they're neat, and I want to share with you why I think they're neat. How does that sound? So obviously this is not going to be any kind of comprehensive list, and there's not even a distinct range of time that I'm pulling from here. This is just cool stuff that I saw, and I want to give my opinion on it, but I don't think it's one of those things that we need to go deep into and dissect the methodology of the paper. Not that that's not important, it's just there's a lot of papers I want to get through, and I don't have a lot of time, so I just wanted to touch on them and say, yo, look at this cool finding. This is why this is important. This is where it fits in the grand scheme of the arguments regarding that topic. Let's move on. So let's get some honorable mentions out of the way right off the bat. Honorable mention number one is going to be the 100 plus page preprint that is currently available by Lieberger and his team on Homo naledi. For those of you who might be unaware, there's a hominin called Homo naledi. It's from South Africa, and the team that discovered it has made some pretty big claims lately that Homo naledi was getting back into the inner recesses of this cave system, bringing their dead with them, digging holes and burying them intentionally in there. This is a big deal for a few reasons, but the main one is that Homo naledi is extremely primitive looking. In a lot of ways, this thing looks more like Australopithecus than it does late members of genus Homo, like us, Homo sapiens, for instance. And in fact, there was a poster presentation here out of the European Society for the Study of Human Evolution, a conference much like the AABAs that I go to every year and tell you guys about it every time I go, that is making the argument that it might be the case that Homo naledi is actually a relict Australopith, whether that's coming from, genus Australopithecus or whether it's, a member of whatever weirdly gracile Paranthropus. They don't really make a case that it's either one in particular, but it's something that people are clearly still considering. Now, my understanding is that Homo naledi, while it has a lot of autapomorphic characteristics that are specific just to Homo naledi, also has a lot of traits both craniodentally and postcranially that align it with genus Homo. So I'm going to be interested to see if this poster presentation ends up making it as like a published paper somewhere, so we can really dig into that and see if they make their case. Because as of right now, I'm pretty convinced that Homo naledi is a member of genus Homo, but again, we'll see. The controversies never end with this thing. Of course, this 161 page, I just checked, preprint is not concerned with a poster presentation from the European Society for the Study of Human Evolution. It's more concerned with trying to remake the case, or at least make the case a little bit more comprehensively, that Homo naledi was actually burying its dead. There's a lot of controversy about this. A lot of people don't think that Homo naledi was burying its dead down there, but at the same time, it is the only thing in that cave virtually. There's nothing else in there other than some baboon remains, essentially. So there's a lot of back and forth going on in the scientific community as to whether or not Homo naledi did in fact bury its dead. I've covered it extensively on this channel. The TLDR is that a lot of people who specialize in burials don't find this case compelling, but Lieberger and his team have like I just said, published a recent preprint that is like, doubling down, really it's more like a quadruple down on this idea that Homo naledi was in fact burying its dead. And the response of the scientific community to this new preprint is unknown because it's 100 pages long. No one's had time to read it yet. I'm still working through it, and a lot of people who have been consistently covering this topic here on YouTube are also still working through it. You're gonna hear from me one way or another on the subject, but so far my impression is that it's a lot of the same. Still, we'll see. The next honorable mention comes from Andrew Barr and Bernard Wood, who did a fantastic sort of summary paper on the potential bias in hominin sampling. Basically, and you guys probably know this if you've spent any modicum of time here on my channel, but we suppose that hominin evolution, so the evolution of our lineage after the split with panins, our closest living relatives, the chimps and the bonobos, we suppose that most of that happened in Eastern Africa and in Southern Africa. And the reason for that is because that is where we find all of the hominin fossils. But there is a problem here. That's where we find all of the hominin fossils because that is where we are looking. A lot of this research started because folks who were quarrying for other important minerals and resources kind of stumbled across like hominin remains and were essentially like, oh my gosh, like there's human relatives here, we should look for more. And that's where these modern research projects have kind of bloomed from. It's where we can trace the roots of those projects. The most classic example of this is the Tong child. And the reason I say it's the most classic is it's because it's one of the first examples. Essentially, there was a lime quarry down in South Africa, and quarrymen discovered the Tong child. It's the cranium of a little Australopithecus africanus, and it was the first member of Australopithecus ever found. The art described it and was like, oh my gosh, this is gonna shake everything up, because it told us, first of all, that hominins probably evolved in Africa and not in Europe, specifically the UK, which is what a lot of people were kicking around at the time. And it showed us that probably bipedalism evolved before big brains. But you can see how this works. We find the Tong child in South Africa, and so people start looking for more remains in South Africa. Similar stuff going on in East Africa, and it ends up creating this massive bias because who is to say that hominins were only in those two locations? Barr and Wood make a very apt and interesting comparison by looking at baboon diversity. They point to the fact that we have baboons in the Rift Valley, specifically in the Eastern Rift Valley in Eastern Africa, and we also have baboons absolutely everywhere else on continental Africa. And that's important because baboons, like early hominins, are generalists. They're capable of living in all sorts of different niches. And so Barr and Wood point out that if we are to use baboon diversity just in Eastern Africa to represent total baboon diversity, we would be drastically underestimating it, whether we're talking about behavior, morphology, or obviously geographic range. Obviously, it's probably time to pick up the shovels and look a little bit more in Central and Western Africa in areas that are potentially fossiliferous, but the big question is, where do you start? And that's also all assuming that the comparison between modern baboon diversity and ancient early hominin diversity is an appropriate comparison to make. I think it probably is, but we should probably look a little bit more into it just to be safe. Our next honorable mention comes from hominin colleagues who were investigating the effect of an incomplete fossil record on our ability to track and understand evolution within those strata. This paper is pretty cool, although it's a little bit outside of my normal realm of expertise, so, forgive me if I bungle anything here. My understanding is that they simulated various tempos of evolution, so stasis versus directional selection, but gradualistically, and then they simulated an incomplete fossil record by knocking out various steps along the way to see if using the incomplete fossil record, we could accurately reconstruct the known simulated tempo of evolution. The main reason why this is an important question to ask is obviously for paleontological and scientific reasons, but what's cool about this paper is that this is answering a question that Darwin had asked all the way when he first published Origin in the late 1850s, and it turns out the answer is yes, you can correctly suss out the mode of evolution even with irregular age depth models. Neat. The last honorable mention comes from Lewin and colleagues, and it has to do with human evolution in kind of a roundabout way, much like the last one. It's actually on the nature of the scrambled genomes found within annelids, or worms. Basically, these guys sequenced the genomes of a lot of different worms, or annelids. You can think of like earthworms and also their close relatives like leeches or bloodworms or whatever. And what they found is that their genomic structure varies a lot in between different species, even closely related ones. So not the sequence identity or what the genome is actually saying, but the structure. Everything is swapped around and shifted and scrambled, if you will. And what they found is that that scrambling can happen in a stepwise fashion, or it can happen like super quickly. To me, this has bearing on human evolution because it explains a lot of what we see in the Y chromosomal difference between hominins and pannins. So humans and chimpanzees, despite being ridiculously genomically similar, 98.8% when looking at the coding base pairs and roughly 96% when looking at the whole genome, we're more similar to them than rats are to mice, for instance. But the Y chromosome in particular is like notoriously very different between hominins and pannins. And in fact, a recent paper outlined that the Y chromosome is actually very different between all the great apes, and another recent paper talked about how Y chromosomal evolution is really fast in primates in general, so it's not just a great ape thing. But the reason I'm bringing up this Annelid paper is that it shows how quickly you can have the scrambling of an entire genome. In the face of ridiculously closely related worms having completely rearranged genomes with respect to one another, a slightly rearranged Y chromosome in pannins compared to hominins just is even less of a deal than it was before. And it tells us that the rearrangement of entire genomes really isn't all that uncommon, let alone the rearrangements within individual chromosomes. Okay, so let's talk about the big boys, the top papers from the recent past that I want to talk about. These are going to be in no particular order of coolness, but number 4 has got to be Vocal Labeling of Others by Non-Human Primates by Oren and colleagues, and it concerns some of my most favorite primates, marmosets. Marmosets are platyrrhines. They are New World monkeys, so they're found in Central and South America, and they're really unique for a couple of different reasons. First, their social system is really weird. Like humans, they tend to exhibit a lot of pair bonding or monogamy, but also like humans, sometimes they live in little marmoset polycules where they have one breeding male and a handful of breeding females, or one breeding female and a handful of breeding males, or a mixture, but they always live in little small groups. Usually it's a breeding pair and their offspring, but marmosets are interesting, they're flexible, and I think they're cute. They're like little cat primates. Which brings me to the second thing that's cool about marmosets, which is that they've secondarily re-evolved claws. Now, these little nails, these tegulae, can be utilized in their pursuit of gums along with their specialized incisors. So they're gum feeders, and sometimes they eat like insects, classic primate behavior, opportunistic omnivory. And one of the last things that I love about marmosets is they'd have a really cool communication system. Their little calls and trills are kind of unique to them, and that's made them a lovely study species for research into primate communication and perhaps some of the basest origins of language. Marmosets aren't known for being the most cerebral of the primates. They're not known particularly for their cognition. In fact, their brains are quite smooth in the literal sense, unless you insert the human-specific RGAP11B into them at the fetal stages, then they'll develop a much larger brain, which is kind of freaky. They did that back in 2020, and I made a video on it back then. But we've probably underestimated them, because it turns out that marmosets label one another with individual specific calls, like names, something that was previously only thought to be done by humans, dolphins, and elephants. In fact, not only are these marmosets labeling their conspecifics, but they also respond to specific labels when other conspecifics use them on them. And specifically, there were similarities between the calls used to label specific individuals within families as opposed to unrelated individuals, meaning that within a family of marmosets, each individual of the family might refer to a specific marmoset using very similar sounding calls, which, to anthropomorphize, sounds a lot like a name that everyone has agreed on. This study is not just important because it's another instance of like specific referral calls for individuals, names, and I put a heavy asterisk on that. But because it's within primates, within our order, right? And because it's specifically within a species that is not known for being highly cerebral. This suggests that the basis for naming and for specifically labeling individuals with specific calls or vocalizations, that might be a much more primitive feature than we previously thought. More and more, we seem to be finding that the basis for language, that stage, was set a lot more distantly in the past than we had, I guess, previously appreciated. Because we like to characterize language as like, okay, it's this uniquely human thing. And as we see it today, it kind of is. But because we keep finding the steps along the way to the human condition in other primates, some of which we've previously considered like not very cognitive, I think it's safe to say that language as a categorical difference is pretty much a dead idea. Steps in evolution are rarely de novo. Typically, there's a precursor, so it won't surprise me if we continue to sort of erode away at this idea of language as this uniquely human conception, even considering extant primates that we share the world with currently. Number 3, of course, has got to be the brand new Australopithecus anamensis mandible by Lewis and colleagues that's come out of Ileret, and this one's important for a pretty straightforward reason. On paper and under standard and traditional conventions, this mandible makes it very, very difficult to propose that genus Ardipithecus gave rise to genus Australopithecus. Two terms that you probably remember from biology in high school are anagenesis versus cladogenesis. The former is when you have a population of organisms that slowly, gradually evolves into a different population. This is speciation on a single lineage, whereas cladogenesis has an ancestral population, and from that ancestral population, 2 new populations descend that are distinct species. The ultimate example of anagenesis is perhaps the famously outdated and maligned March of Progress, which virtually zero biological anthropologist holds to today, except in very specific and limited circumstances. Most evolution is better described as like a bush of life, where we have a lot of cladogenetic events and a lot of incomplete lineage sorting and gene flow. Part of the reason why cladogenesis is favored over anagenesis within human evolution is because we just have so many different species of hominin, and it's very difficult to definitively say which population gives rise to which subsequent population. Now, conventionally speaking, within the fossil record, within paleontology, there is one decent way to preclude anagenesis, again, at least according to conventional methods, and that's if you suddenly find that a proposed descendant is living contemporaneously with a proposed ancestor. The idea is that if they are living at the same time, the ancestor shouldn't be considered the descendant because anagenetically that doesn't make sense, and also cladogenetically that doesn't make sense. Now, I know what you're thinking. It's probably something that I have mentioned before on this channel, but at least with extant populations, that preclusion isn't necessarily appropriate. This is for the very simple reason that, as we see today, when a speciation event happens and a new species forms from a pre-existing species, it descends from a pre-existing species, that pre-existing species doesn't have to go extinct. And my favorite example of this is dogs descend from wolves, and we still have wolves. Officially, this is called peripatric speciation, and while, again, this is reasonable to propose for extant species, because we see it, it's very difficult to apply to the fossil record, and so it's common practice, for the sake of standardizability, to just say if they're contemporaneous, then one didn't descend from the other, rather they both descend from a parent population that perhaps was very similar to this previously proposed ancestor. When Ardipithecus ramidus was discovered in the 1990s and published on in 2009, it was proposed to be a potential ancestral genus to Australopithecus. Specifically, the proposed anagenetic relationship was Ardipithecus ramidus at 4.4 million years ago, Australopithecus anamensis at approximately 4 million years ago, and Australopithecus afarensis lucy species significantly later at approximately 3.5 million years ago. Now, the problems abounded almost immediately because we have older Australopithecus afarensis than 3.5 million years ago, and there was already coming to the surface Australopithecus anamensis remains older than 4 million years ago. But we didn't have anything that was 4.4 million years ago from any Australopithecus, which meant that Ardipithecus ramidus was still safe as this potential ancestral genus, even if it was cutting it pretty close. After all, even assuming that Australopithecus anamensis is 4 million years ago and Ardipithecus ramidus is 4.4 million years ago, there's a lot of change that has to happen in just 400,000 years. Ardipithecus, compared to Australopithecus, has larger canines, it's less sexually dimorphic, and the biggest thing is that it's less committed to terrestrial bipedality. Ardipithecus ramidus still has a host of suites that allow it to get around in the trees and indicate that it wasn't like spending the majority of its life on the ground walking around bipedally, unlike Australopithecus as a genus. The implications of this new mandible from Australopithecus anamensis at 4.3 million years of age, a mere 100,000 years younger than Ardipithecus ramidus, should be immediately obvious. You're not going to get like all of the morphologic change between Ardipithecus ramidus to Australopithecus anamensis in 100,000 years. I mean, the foot alone requires a complete restructuring. But in addition to that, we're talking about large-scale morphologic change across the body of this animal, from the pelvis to the teeth. Basically, the point is, if we're being standard and conventional, this mandible from Ileret makes it really, really unlikely that Ardipithecus ramidus is ancestral to genus Australopithecus. And what that would mean is that Ardipithecus ramidus, at least, is almost definitely not in the direct lineage leading to Homo sapiens, and is probably a cousin to Australopithecus in the same way that Paranthropus is. But put an asterisk there. Because, again, it's possible that peripatric speciation was at play here, and that Ardipithecus ramidus emerged initially earlier in time than 4.4 million years ago by a lot, and that a population budded off from Ardipithecus ramidus in the more distant past, and that population became genus Australopithecus, with Ardipithecus ramidus still living and persisting to eventually be contemporaneous with what would become its descendant. Or it could be that Ardipithecus ramidus isn't ancestral to Australopithecus, but Ardipithecus the genus still is, in the form of something like Ardipithecus cadaba. This argument is going to continue to rage for quite some time, especially because technically there still is 100,000 years for Ardipithecus ramidus to be the ancestor of Australopithecus anamensis, even using conventional definitions. So we'll see how this one ends up hashing out. To be clear, none of this has any impact on whether or not Ardipithecus ramidus is a hominin or whether Ardipithecus the genus belongs to the hominin group. All this would really do is shake up how we look at the family tree a little bit, which is par for the course in biological anthropology, I would say. Number 2 is the early evolution of small body size in Homo floresiensis by Kaifu and colleagues. And this paper is predicated on some new postcranial remains from Homo floresiensis, the approximately 3-foot tall hobbit hominin from the island of Flores. These new remains are interesting for a whole boatload of reasons, so let's go through those briefly. The first is that these remains are from a site that's dated to, on the older end, more than 700,000 years ago. This is pretty critical because most of our Homo floresiensis remains are younger than 100,000 years. This means that this little guy has been on this little island for quite some time. The second reason is that the postcranial remains are very little. They are little even compared to the more recent Homo floresiensis remains. Specifically, they're 9 to 16% shorter or thinner than the more modern Homo floresiensis. So it seems like these guys as a lineage got really small and then actually got like a tiny bit bigger as we move into the more recent past. That makes me wonder if they were already small before they got to Flores, given that's, 600,000 years older than what we've been looking at from Homo floresiensis, and they've been small that entire time. How many islands did Homo floresiensis colonize before Flores? If they did, in fact, colonize other islands before Flores, how long have they been small? What do they actually descend from? And that's an interesting question that this paper also has thoughts on. They propose that Homo floresiensis descends from an Asian lineage of Homo erectus. This is pretty typical. This is the idea that was initially put forth when the first Homo floresiensis remains came to light. It came basically from an overall morphologic comparison of Homo floresiensis to members of genus Homo, and this holds with the current study, looking specifically at aspects of the humerus. The humerus seems to look more like Asian Homo erectus than any other member of genus Homo. However, the competing hypothesis to this is that there are aspects of Homo floresiensis that don't look like Homo erectus. They actually look like Australopithecus, namely aspects of the carpals or the wrist bones and of the pelvis. Evidently, that relationship also holds with the recent European Society for the Study of Human Evolution poster presentation that looked at the sulcal patterns of the LB1 or primary Homo floresiensis skull that we have, endocast, which also seems to link it more with Australopithecus than with Homo. Again, we'll have to see what comes from some of these poster presentations if they end up getting published in journals, but right now, the ancestry of Homo floresiensis is really up for debate. I don't think that anybody is suggesting that it descends directly from Australopithecus, but rather that it's probably a line really, really closely after the emergence of genus Homo, because if it does descend directly from Australopithecus, that means it's probably not genus Homo. It would make genus Homo non-monophyletic, which you can't have. That's a big no-no. Homo floresiensis is such a fun grab bag mixture of characteristics from genus Homo and from Australopithecus, and it's such a long-lived species as well, from at least 700,000 years ago to the pretty recent past, within the last 100,000 years. I'm psyched to see where things end up with it. It's good to know that we're still pulling up remains from Homo floresiensis that may elucidate things a little bit further still. And number one, our briefest foray is on a brand new Neanderthal genome, and it's from a paper that's titled Long Genetic and Social Isolation of Neanderthals Before Their Extinction. This paper, like I said, sequenced a new genome of a Neanderthal that they've nicknamed Thorin, and what they found is that Thorin belonged to a population of Neanderthals that was ridiculously isolated from other populations of Neanderthals, as in they didn't have any gene flow, they didn't interbreed with one another. And this is despite the fact that geographically they weren't isolated at all. In fact, there was a neighboring group of Neanderthals within a 10-day walk, and yet for tens of thousands of years, this specific population had no interbreeding with them. The specifics that the paper gives is that this group of Neanderthals split from all other Neanderthals roughly 105,000 years ago, and given Neanderthals went extinct roughly 40,000 years ago, we're looking at over 50,000 years of genetic isolation, within this group. This might imply some interesting social proclivities for Homo neanderthalensis, right? Why did this group stay isolated genomically? Why wouldn't they interbreed with other groups, seemingly? This area has a rich archaeological history, and it's not in the middle of nowhere tucked away. It's right on the beaten path. If Neanderthals are super picky over who they will interbreed with, this might have something to say about why they're extinct and why we're not. An already inbred population that is isolating and further inbreeding still isn't going to be the most adaptable, in my opinion. Anyways, my gentle and of course very modern apes, this is, as always, an exciting time to be interested in science and in biological anthropology. I just wanted to share with you a couple of passing thoughts that I had that are really, not the most groundbreaking, if I'm being perfectly honest. I'm just trying to sort of contextualize these finds within, what I know generally about biological anthropology and how it's furthering the conversation. So I hope that's been interesting for you. It's been fun for me. If you like what I do, please consider supporting the channel in the free way, which is, liking, commenting, subscribing. If you can't think of what to comment, tell me your favorite recent paper. I'd love to check it out. And if you want to support me in a more financial way, you can join my Patreon. We You can join my Patreon. Patrons get their names at the end of every video, and when I'm on top of things, they get early access to videos. I can tell you right now, they are going to have early access to this big documentary that I'm working on. So if that's something that is enticing to you, consider throwing me a couple of bucks. And in the meantime, I want to thank you from the bottom of my gentle and of course very modern heart for being such a curious primate, and I'll see you next time.